Hey y'all, in this video, I'm going to do a real quick review and recap of some of the concepts in mitosis and meiosis that can be a little tricky for students. In this example, I'm going to use the human cells with 46 chromosomes to go through the two processes and compare how the DNA is split. To start with, prior to mitosis and meiosis, during interphase is the S phase, which is when the DNA gets copied. So that at the end of the S phase, you end up with 92 chromatids. And it's these 92 chromatids that are going to get split in the processes of mitosis or meiosis, depending on the type of cell that is dividing. So let's start with the process of mitosis. In mitosis, the chromosomes are going to line up during metaphase. And when they line up, they do so along what is known as the metaphase plate. Now, in the case of mitosis, there are homologous chromosomes. For example, we're going to say this one is homologous to this one. So both of these have at this loci the gene for a certain characteristic, let's say hair color. And so I'm going to label these as a homologous pair by simply using letters. So those two are a homologous pair. They both code for the same trait, and you inherit one from your mother and one from your father. But they do not necessarily line up side by side in mitosis. In mitosis, all the chromosomes are lined up at the metaphase plate. There's another homologous pair, and there's a third homologous pair. Now, what happens in mitosis is during anaphase, the microtubules, which have been connected to the kinetochores of these chromosomes, start to pull the chromosomes apart. And so during anaphase, the microtubules, which are attached to kinetochores, proteins, located right at the centromere of these chromosomes, the microtubules begin to shorten and pull the sister chromatids apart in opposite directions. And the result of the sister chromatids being pulled apart are two identical cells that are both diploid. Now, in the case of meiosis, there are two rounds of division. But prior to meiosis, there are still 92 chromatids that are going to need to be separated. So let's split this process into meiosis 1 and meiosis 2. So in meiosis 1, it's going to start completely different than mitosis because the homologous pairs do line up along that metaphase plate. So this couple right here is a homologous pair. And they both encode for the same genetic trait: eye color, hair color, the presence of dimples. The next homologous pair lines up as well, and so does the next homologous pair. And that's because in meiosis 1, more specifically in prophase, there's an important event that's going to happen called crossing over, which is the exchange of genetic information. At the end of meiosis 1, the cells are going to split by splitting the homologous chromosomes first. So again, microtubules are responsible for pulling at the kinetochores of these chromosomes so that what you have at the end of meiosis 1 are two diploid daughter cells that have one each of the homologous pairs. Right at the end of meiosis 1, there's no interphase and thus no S phase for the DNA to get copied again. Instead, the cells, the two daughter cells, are going to go immediately into meiosis 2. And during meiosis 2, we are now going to pull apart the sister chromatids of the chromosomes in each daughter cell. And what you have left are 4 daughter cells that are all haploid. Now, if crossing over does not take place, these two will be identical to each other, and these two will be identical to each other. However, if crossing over does take place, then the result are 4 genetically different cells. Now, in the case of humans, this is a little bit different because of the number of chromosomes that we have. The basic understanding for humans is that when crossing over does take place, the genetic diversity of the offspring will increase. And another factor that's going to increase the genetic diversity of the offspring is not only just crossing over, but the fact that it is fertilization, in other words, an egg and a sperm from a completely different donor combine to make the offspring neither a copy of either parent, but instead both sets of genes combined. I hope this video was helpful. Thanks for watching. Bye.